Hello everyone and welcome to our third lecture. Today we will be talking a bit more about microscopy, going into more details about how these systems developed and what you can see using different types of microscope systems and how they actually work. So just a quick definition here that microscopy is the field using microscopes to view objects that are too small to be seen with your naked eye, to be seen with the unaided eye. They are generally separated into three known branches. These are your optical microscopes. So optical microscopes are also referred to as a light microscope. It uses visible light and a system of lenses to generate a magnified image of small objects. This obviously includes microbes, bacteria, fungi, and the like. Then we move on to electron microscope. So electron microscope is a microscope that uses a beam of accelerated electrons as a source of illumination. So something to keep in mind here, as the wavelengths of these electrons can be much shorter than that of visible light photons, electron microscopes can have a much higher resolving power than light microscopes and can reveal the structure of smaller objects. Then we go on to scanning probes or scanning microscopy. So scanning probe microscopy, also abbreviated as SPM, is a branch of microscopy that forms images on surfaces using a physical probe that scans the specimen. So this image down here that you guys can see, this was actually taken by a SPM or a scanning probe microscope. This is an image of what pollen looks like. So for all of you allergy sufferers, this is what gets you every spring. Moving on, there's currently a emerging branch. It's been emerging for a couple of years, so I don't know that the term emerging is necessarily the correct term. That is X-ray microscopy. So as the name suggests, it uses electromagnetic radiation, specifically in the soft X-ray band, to produce magnified images of objects. Since these X-rays penetrate most objects, there's no need to specifically prepare them for X-ray microscopy observations. So this obviously constitutes quite a big advantage of this new branch of microscopy. Moving on, so first, the first bunch of slides that we will be talking about will all be dealing with bright field microscopy. So what is bright field? A bright field microscope is also known as a compound light microscope. This is an optical microscope that uses light rays to produce a dark image against a bright background, hence bright field. So this image here, this is actually an image of tissue paper. You can see the threads and the strands appearing dark against the light or the bright background, and that's where this type of microscope gets its name. This is a standard microscope. You guys will be using them, and that is used in biology, cellular biology, microbiological studies. So those of you signed up for our practical sessions, you will be using bright field microscopy for the most part. Then moving, just a little throwback here to one of our previous lectures. You guys remember Antoni van Leeuwenhoek with his, the guy that owned those many small microscopes. He developed essentially simple microscopes. So these microscopes only had one lens. You can see that over there. And remember, he used these microscopes, this collection of microscopes, to view what he called animalcules. So this is just a throwback to that, just to serve as a bit of a reminder for you guys. Now, moving on to what you guys might think of when you think about a contemporary microscope. This is what is known as a compound microscope. So a compound microscope is a high power or high magnification microscope that uses a compound lens system. So as the name suggests, with compound, it means constituting more than one part. So the compound microscope has multiple lenses. Specifically, it usually has an objective lens, which you can see down here. Those are your objective lenses. And then it is compounded or multiplied by the eyepiece or the ocular lenses. So as a typical, just a simple example, if you have an eyepiece lens that is at 10 times magnification and you have an objective lens that's also at 10 times magnification, that will result in 100-fold increase in size. So the higher magnification in these types of microscopes is achieved by using two lenses rather than just a single one, like that one on the previous slide that we just looked at. So while the eyepieces and the objective lenses create a high magnification, a condenser beneath the stage, here you can see the condenser over there, focuses light directly onto the samples. Then we will be talking obviously now about resolving power or resolution. So what is the resolution of a microscope? Well, the resolution of an optical microscope is defined as the shortest distance between 2 points on a specimen that can still be distinguished by an observer or camera system as separate in entities. So essentially, what is the distance that you can still see two things as two things where they don't meld together as one blob? You can see here, the resolution allows us to see these objects here as separate from one another. Another term that I want you guys to be familiar with is useful magnification. What useful magnification refers to, over here you can see it. It means it's an improvement in your resolution with greater magnification. However, there's an important limit that you guys are going to have to try and keep up here. So in order to observe a specimen with fine details in an optical microscope, the minute features present must be sufficient to contrast and project an intermediate image at an angle that is somewhat larger than the angular resolving power of the human eye. So at a selected numerical aperture, when the microscope provides a magnified image that has a magnitude equal to the resolution limit of the human eye, additional magnification beyond this point does not result in resolving or even smaller specimen detail. So I know that's a pretty complex, or it sounds pretty convoluted, but essentially what it means is there's no point in trying to have the resolution or the exceed what your eye is actually capable of perceiving. So the range of useful magnification for an objective or eyepiece combination is defined by the numerical aperture. You're definitely going to have to be familiar with that term, numerical aperture. So there is a minimum magnification necessary for detail present in an image to be resolved, and this value is set rather arbitrarily as 500 times the numerical aperture. And then it's defined by an equation that we will be going into maybe a bit later. So the take-home message from everything that I just said is essentially that the range of useful magnification for an objective and eyepiece combination, so the total magnification, is defined by the numerical aperture of the microscope system. So if we look here just as a summary here, the minimum distance between two resolvable points, that is what your resolution or your resolving power will describe. And then another factor that I want you guys to keep in mind here is that the wavelength of the light is also a major factor. So if you look at the visible spectrum, moving from your purples and to your reds, you have an increase in wavelength of the photon wavelengths. So a visible light then varies from this 400 to 700 nanometers with smaller wavelengths, giving you better resolution. So it is better to use light on this side of the spectrum than on this side of the spectrum. Again, here you can see what there's a resolution of a microscope will allow you to do. Here the points are unresolved. Here they become partially resolved, and here you can see them nicely as two distinct dots. So something that I want you guys to definitely practice in your own time. You can look for examples in your textbook, or failing that, you can definitely find some online. I know a lot of you guys are not familiar with the metric system, or maybe not as comfortable with the metric system, but as scientists, you are expected to know how to convert units. So you have to be able to tell me that 50 nanometers is the same as 0.05 micrometers, or 800 micrometers is the same as 0.8 millimeters. So you have to be able to convert freely between these units In exams or tests, I might ask you to do that, or I might provide you with units of specific, using a specific measurement style, and you will need to make sure that they're all uniform, and thus you'll need to be able to convert between these different measurement lengths. So just as a tip here, always remember that when you're going from a smaller unit to a larger, the decimal moves to the left, and when you're going from a larger to a smaller, the decimal will move to the right. It is pretty simple because all of them work in tens. So as long as you know that, 1 millimeter is equal to 0.01 centimeters, and for all the other ones, you should also be able to simply do these conversions. Okay, so getting back to our useful magnifications. So just as a reminder, remember that the range of useful magnification for an objective or eyepiece combination is defined by the numerical aperture of the microscope system. So that obviously begs the question, so what exactly is this numerical aperture? What does that actually mean? So what a numerical aperture is, it's the range of angles over which a system can accept or emit light. The numerical aperture of a microscope is the measure of its ability to gather light and then resolve the fine specimen while working at a fixed object distance. So if you look at this equation down here, just a couple of definitions here, because that's how you can calculate your numerical aperture. This micro symbol that you see over here equals 1/2 of the objective's opening angle. You can see it over there. Your angle is half, so there's your micro refers to that. And then your n in your at the start of your equation is the refractive index of the image medium between the front lens of the objective and the specimen cover glass. This value typically ranges from 1 for air to 1.51 for specialized immersion oils. You don't have to memorize all their refractive indexes. I will provide them if I ask you a question on this. Some things to keep in mind, though, that the smaller the object, the more pronounced the diffraction of incident light rays will be. Higher values of numerical aperture permit increasingly oblique rays to enter the microscope, the objective front lens, which produce a more highly resolved image and allow smaller structures to be visualized with higher clarity. So this all just deals with how much of the light that's being reflected by the organism are you actually able to capture and then use to distinguish the details within the organism. So in order to increase the effective aperture and resolving power of a microscope, a condenser is typically added. Remember, I showed you on that sketch the condenser below the specimen plate. And this generates a ray cone on the illumination side of the specimen. This enables the objective to gather light rays that are the result of larger diffraction angles, increasing the resolution of the microscope system. So the light source, the location of the light source is purposefully there so that we catch more of the light coming from the specimen so that we can see more detail. And then the sum of the aperture angles of the objective and the condenser is referred to as the working aperture. If the condenser aperture angle matches the objective, maximum resolution is obtained. I wouldn't break your heads about this too much. Go through this portion of text in your books. Make sure you understand the general take-home message of how the light is captured, that you want as much light captured as possible so that you can distinguish the detail, and that refraction, in this case, losing light, would be very, very bad. So moving on to our next definition of depth of field. You'll often find this in when you work with microscopes. So what is depth of field? So depth of field is simply the measurement of the amount of distance between the nearest and furthest objects that appear in acceptably sharp focus. So if you look here at this image over here, you can see that the red fiber is in very, very good focus, yet the blue and the yellow are not. And that is a function of your depth of field. Your focal point in this case is exactly on the red fiber. Your red thread is in focus, but the depth of field is too shallow to capture all three threads in sharp focus. Then another thing that you want to keep in mind is your area of field. This is much easier to understand. This is just the area that you are capturing. So that would be 1.5 millimeter squared in this example to 0.35 millimeter squared. So it's just how much of an area, how big is the area that you are actually looking at. Now, moving on to how magnification works. Magnification in compound microscopes, pretty simple. You have your objective lens. This is the one that is closest to the specimen, the one that's pointing at the specimen that's on the slide. So if it has a magnification in this example of 4 times, so that's how big it makes your original specimen. If your specimen is this dot, that's what the objective lens does to it. Then the ocular will take that and times that by 10 more. That's your total magnification is 40 times. Obviously, these dots are not to scale. It's just to illustrate the concept here for you guys. So that's how you view total magnification of a microscope. Just as a bit of a review here, I want you to show you guys that higher magnification will result in a high NA. Remember your numerical aperture, C. As your magnification increases, your numerical aperture will also increase. It results in a stronger resolution power, so a strong resolution. So there's a small distance. You can distinguish detail over very, very small distances. So this is very good. Sorry, these seem to be pretty badly aligned here. And then a shallow depth of field. So obviously, the more you are zooming into your sample, you're losing some of the detail above and below it. That's what this number illustrates over here. You have a smaller area of field also, obviously. Your depth of field is represented by, this value over here. Your resolving power is, so it isn't badly lined up. Sorry, I mixed that up. Your resolving power is over here, where smaller distances for resolving power is in fact good, because you want to be able to distinguish between small distances. Your depth of field will also decrease. This is less ideal because now you're losing some of the detail on the top and the bottom. Remember the 3 fibers. And then your area of field, obviously, as you're essentially zooming in or focusing in smaller and smaller and smaller, the entire area, the total area that you're viewing is actually decreasing in size. So yeah. So how do we deal with the problem of the refraction? So think about it like this. What happens when light moves from one medium to another? So in this case, from your glass slide to the air and then to the lens. It's similar to when you're looking at a pool of water. So when light moves from one media to another, its angle it changes due to refraction. Sorry. So if you look at this, this is what this line here describes as this. The light from the light source down here leaves the glass slide. It becomes refracted, so it's missing this lane. So you're not seeing any of that light. This is a problem. So how do you deal with that? That is where oil immersion comes in. So oil immersion creates a smaller refractive difference, so refraction angle, so you're not losing that light. The oil then lets you view more of the detail. It allows more of the light from the light source to pass through the sample and actually being viewed as opposed to just not being viewed. So it creates more detail and you can see just better. It increases both your resolution and numerical aperture. Okay. So all of that, what we just discussed, all deals with bright field microscopy. So looking now, there are other types of microscopy that also relies on light. So they have similar principles that apply. This includes dark field microscopy. So as the name suggests, you can already imagine how different it'll look to light field, where the field will now be dark and the specimen will be light. Phase contrast microscopy. We will be looking at examples of all of these. Fluorescence microscopy, all of them rely on light. And then there are 2 types of microscopes we will be discussing that do not rely on light. Remember the electrons we spoke about. That includes transmission electron microscopy, that's TEM, or scanning electron microscopy, that's SEM. So how does dark field look? Remember, I told you, bright field, think of the white background, hence where it gets its name, the bright background. So it's a bright field that's surrounding your organism or surrounding your sample, where with dark field, the background is completely black, it's dark. So in this instance, light is directed at a sample at an angle, and the light scattered from the specimen is what is observed. Most of the light directed at the object is not received by the microscope, only the light that is refracted by the specimen. So think of it this way, it's essentially almost, you can think of it as similar as like how a mirror functions, where it's the light that's coming back that you are actually viewing. So the light that hits the specimen and then bounces back to you, that's what you're seeing. And that's why the specimen appears, yet the light that just passes through the field and misses your specimen appears black, because it doesn't get reflected, it doesn't get refracted back to you. So this type of microscopy is frequently used to examine live specimens or structures that can be damaged if you were to stain them for bright field, for the bright field process. So it's more sensitive in that sense, or it can be used to examine more sensitive structures, I should say, sensitive structures and specimens that could be disrupted or damaged by staining. So, okay, moving on to phase contrast microscopy. So this is sort of, you can think of this sort of as like a combination between bright and dark field. It brings together two sets of light rays, direct rays, direct rays and diffracted rays to form an image. And it allows for an examination of living organisms and all internal cell structures. And these internal cell structures will be very finely and sharply defined. So yeah, you can see here just how different that looks to what we were looking at now. It's kind of like almost, you can view it as a best of both worlds type of microscopy technique. So here you can see the looking at yeast cells using bright field over here, phase contrast, and dark field microscopy. See how this sort of looks like it's almost a hybrid of the two, because it relies on both the refracted light and direct illumination. So yeah, it uses both, and that's why it produces images like that. And see, notably here, how much more focus there is on the internal structure. You can see more detail there than you can there or there. Okay, moving on to fluorescence microscopy. So I suppose we'll start with what makes these things fluoresce. So fluorescent molecules contain fluorophores. And a fluorophore is something that absorbs light energy at a specific wavelength and then re-emits it at a longer wavelength. So it'll take, here you can see the excitation emission spectrum down here for fluorophore, that is its chemical, what it looks like. So this would be the excitation wavelength. So this is the wavelength that's hitting the fluorophore, and then this is the emission. This is what the fluorophore then pushes back out. That's not the right word. Re-emits, sorry, that's the word. So that's what it re-emits at a longer wavelength. Specifically, you look at the change in wavelength, where it's the short wavelength that comes in and the long wavelength that goes out. So very popularly, you can see the green structures here. So it's green fluorescent protein is a very popular way to visualize structures. GFP, as it's also known, was originally isolated from a jellyfish. Here you can see the jellyfish. So essentially how you would then look at the structures you want to is you would make a transgenic organism that expresses the genes from the GFP that allows you to visualize the internal structures, as is this example over here. Now, moving back to our friend, the tardigrade, here you can see a good example of all the detail that you can see using fluorescent stains. In this instance, this was actually the image that won the Olympus Global Image of the Year Award. It was by, I'm going to butcher this name, Tagide de Carvalho, who said of this image that they used multiple fluorescent stains and capitalized on the natural fluorescence of the samples. Oh, a lot of tongue twisters there. So these fluorescent dyes allowed them to see the tardigrade's entire digestive tract, including the mouth parts, and then the stomach filled with food. This is really, really interesting. Remember, the tardigrade was that really tough organism we discussed, the one that can go into space and come back and still be okay. The water bear moss piglet, yeah. Okay, so something else that we can use fluorescence for is for the fluorescent antibody technique. So this is a frequently used technique for visualizing cell structures. The fluorescent antibodies essentially here bind to the bacteria on a microscope slide, allowing us to detect the bacteria using a fluorescence microscope. Thus, the technique is valuable for visualizing certain bacteria that are difficult to isolate from culture or patient samples. So here you can see the antibody with the fluorescent dyes attaching to the bacteria. See there, it's nice and attached, and then that's what it would look like under the fluorescent microscope. As I've just stated, this is really helpful if you have a microbe that's difficult to culture or difficult to see otherwise. And how this essentially works is these antibodies match specific proteins on the cell surface that can then be used with the fluorescent dye to see the cell. Okay, something else that fluorescence is often used for is live dead staining or viability staining. So here you use a differential stain to distinguish between live and dead cells. In this example, everything you see that is green is assumed to be alive, and everything that is red is assumed to be dead. The reason being is this, as in the previous example, this dye relies on the cell having an intact cell wall. So the red cells are assumed to have lysed or have a compromised cell wall, thus they're assumed to be dead, and the green ones are still alive. Okay, now moving on to an electron microscopy. I'm not going to go into too great detail here or the technical details. If you are interested in that, please feel free to have a look at your textbooks, but I feel it's a bit above and beyond because you're also not going to be working with these. So just what I want you to know from this is that there are two types of electron microscopes. They both involve sending a stream of electrons at the specimen and either detecting the electrons that pass through, that would be transmission, or bounce off, that would be scanning. And then for the transmission, here you can see the example, see how you can see inside, and that's because it uses very, very, very thin slices to see the internal structures. But importantly, you get no 3D information from this. So the transmission, you can see inside, but you get no idea of the actual 3D shape or structures. With the scanning, you can't see inside because it's just bouncing off the microbe, but you can get an idea of the 3D shape and structure. And here's a nice, again, example of why a TEM is so useful. So a TEM, again, transmission, uses a beam of electron that passes through these ultra-thin sections of a specimen, and then through an electromagnetic lens, then focused on a projector lens. Specimens can be stained, specifically using some heavy metal salts for contrast. But something that's, again, really cool about these microscopes, because they have the insane resolution, far superior to that of compound microscopes. You can see subcellular structures like ribosomes. Here you can see all the arrows pointing at, these are nucleoids. Here are your ribosomes, these little dots. And you can even see the cell wall. You couldn't see that in as great detail using compound microscopes. And then here is a nice example of what a SEM can do for us. This is, again, the scanning electron microscope. So to use the scanning electron microscope, the specimen is coated with a metal, very often gold or a gold palladium alloy. And then an electron gun is used to produce a beam of electron that scans the surface of the entire specimen. Secondary electrons emitted from the specimen produce the three-dimensional image. Here you can see that. This is actually, what do you guys think this is? See, remember when I said this is like little aliens? This is actually an image of a hydrothermal worm. So these are some of the micro organisms that you can find in your hydrothermal vents. Remember, we spoke briefly about those. And this is what they look like. But this thing looks straight like it comes from some alien movie or something that killed a bunch of space marines or something. So colonies of these tube worms live on hydrothermal vents that spew hot mineral-rich water. They are extremophiles, as these places can reach temperatures of 350 degrees Celsius. That, to give you guys a better idea, is 660 degrees Fahrenheit, and these animals live there. Pretty insane. And then something else that we're going to just briefly talk about. This is a atomic force microscopy. So in this instance, as you might guess from the name, you get completely ridiculous resolution. It's also a type of scanning probe microscopy, and the resolution here is in the order of fractions of a nanometer. Remember, that's 10 to the negative 9. That's really, really small. So the picture here is actually of the perfringolysin O toxin from Clostridium perforans. This is the protein that takes hold in the human plasma membranes. And it plays a role in the formation of gas gangrene. Don't look for pictures of that. It's really gross. It really looks really disgusting. People form these, this is gassy bubbles essentially, and their blood turns poisonous, and it's not great. But just the take-home message here is just this atomic force microscopy produces insane resolution. To that end, here we have a brief little summary of the limits of resolution. Here you can see a tick, and with the unaided eye, you can see to about 200 micrometers. Using a simple light microscope, you can get down to about 200 nanometers. Then using a scanning electron and a transmission microscope, sorry, this isn't a simple microscope. This includes compound microscopes, obviously. And then with scanning an electron microscope, that goes down even more, and then the atomic force microscope over here. So I mean, just to give you an idea of scale here, just read this little tip here. So if bacteria is 1 micrometer long and your index finger is 6.5 centimeters long, so just think of how long your index finger actually is, your fingernail. Your entire finger. So how many bacteria could you place into in your finger? The answer is 32,500. Like, we're talking really, really, really small organisms and an insane resolution that these microscopes must have. Okay, so something that we also briefly alluded to now when discussing the microscopes is using staining. So staining helps cells to be clearly viewed, and it allows us to detect structural differences between cells. This can, unstained cells are much harder to see than stained cells, because stained cells have increased contrast. They're best with bright field microscopy. Again, those of you who will be attending the in-person labs, again, I highly recommend, if you feel safe to, please do attend. It's the only way you guys will get the practical experience, the hands-on experience, which is invaluable in this field. It'll also help you with the exams and with the tests, because you will know what you would have done in the lab. You'll remember it a lot better. So it's used often with bright field microscopy, and we will have some of that. You guys will be doing some of that in the labs. And then there are 3 steps. You have to prepare your smear, you have to fix the sample, and then you have to apply the stain. Here is how you prepare your stain. So you spread a thin film of the specimen over a slide with your inoculation loop. You allow it to air dry. You pass it over a flame, and then you flood it with the stain. You rinse it with DI, and then deionized water, and then you look at it using a microscope. So there are different stains that we can do. The first one is just simple staining. You're just using one stain. This is often done just to create some contrast so you can see better, see the organism you're looking at better. Then there are also, so just as I said, the simple stain, you flood the prepare stain with basic, with a basic dye. This is often like crystal violet, saffron, methyl blue, or the carbol. So essentially, you're making it more visible under the microscope, so that it's not just a see-through organism. So how this essentially works, just very basically, basic dyes are positively charged, and the cell surface of bacteria are negatively charged, so opposites attract, and then the cell becomes coated in the stain, and thus you can see the microbe. And then using a differential stain. So in differential staining, you use at least 2 stains that react differently with different types of bacteria, so thus you can use them to separate the bacteria into different groups. The most popular of these, and one you will be doing in the labs, is the classical Gram stain. It is arguably the most famous and most useful differential stain in microbiology. So the Gram stain is useful for the differentiation of, or the separating of bacteria into 2 major groups based on differences in their cell wall. So Gram staining differentiates bacteria by chemical and physical properties of their cell walls. The Gram-positive cells have a thick layer of peptidoglycan in their cell wall that retains the primary stain, crystal violet. Remember, thick layer. And then Gram-negative stains have a thin layer of peptidoglycan that allows the crystal violet to wash out when you add ethanol to it. And then they are stained pink by the counter stain, which is usually saffron. And this technique is usually the first step in identifying a bacterial organism. So here is just what that process actually looks like. You have your bacterial organism, and you stain it first using crystal violet. You use crystal violet because it makes everything purple, and then you add iodine to it. Iodine interacts with the crystal violet to form big complexes known as mordant. Mordant is a chemical that intensifies the stain. Then you add your alcohol. Your alcohol acetone washes away the crystal violet from the Gram-negative cells, but the Gram-positive cells maintain the crystal violet iodine complexes that are trapped within the peptidoglycan cell wall. And you add the counter stain, again, often saffron. So now you will see some of your organisms appearing purple because they retained their initial stain, and some will appear pink. The pink ones are essentially just the colorless ones from the alcohol step. So when you rinse these organisms after they've, the Gram-negative ones, they will lose their coloration because they can't maintain the crystal violet in their cell structure. So thus, when you add the saffron and counter stain, all of them will turn pink. And then you can distinguish between, oh, these 2 are obviously Gram-positive and these 2 are Gram-negative. And this is what it actually looks like. Again, here you can see the pink ones, and there you can see the more purple or dark blue colorish ones. And that's how we can distinguish between them. We also, another stain that's often used is an acid-fast stain. This is a differential stain that's used to identify acid-fast organisms such as members of the genus Mycobacterium. So what is an acid? what are acid-fast organisms? So essentially, an acid-fast organism are characterized by wax-like, nearly impermeable cell walls. They contain mycolic acid and large amounts of fatty acids, waxes, and complex lipids. Acid-fast organisms are highly resistant to disinfectants and dry conditions. And because their cell wall is so resistant to most compounds, they require a special staining technique. The primary stain used in acid-fast staining is carbofuscin, which is lipid soluble. Very important that it's lipid soluble. And apart from being lipid soluble, it also contains phenols, making it aiding in the penetration of the cell wall, which is also further assisted by the addition of heat. That gives you an idea of how resistant these cell walls actually are. that there are so many new steps that need to be included just for us to stain these organisms. So after the addition of heat, the smear is then rinsed with a very strong decolorizer, which strips the stain from all non-acid fast cells, but does not permeate the cell wall of the acid fast organism. So the decolorized non-acid fast cells then take up the counter stain. So here your original stain that you use will be used to identify your acid fast organisms, and the counter stain that you apply will then be used to see organisms that are not acid fast. And here, looking just quickly at special or structural stains, they can be used to identify and study the structure of bacteria. They're useful in observing endospores, capsules, and flagella. So if you look at something like Bacillus cereus, for example, this organism has endospores. Endospores are special structures that allow the cells to survive harsh environmental conditions. More on that in our next lecture or in our next couple of lectures. Yeah, they just create an insane survivability for these organisms. So identifying which organisms are capable of producing endospores is a really helpful thing, and we also use differential stains to do so. So today, just a couple of review questions again, as with the previous lectures. What the minimum distance between two resolvable points is described by is that depth of field, area of field, resolving power, numerical aperture, or none of the above. I'm going to give you guys about 10 seconds or so. I mean, I kind of did go on about this for a bit, so I'm pretty sure all of you will get this. I'll give you about 3 more seconds. And the answer here is, of course, resolving power. Moving on, how does sample immersion in oil aid microscopy? Does it reduce refraction and reflection? Does it bind the sample to the slide? Does it catalyze staining? Does it preserve the sample, or does it do all of the above? Again, I'm going to give you guys about 10 seconds for this. Remember the picture that we looked at. Okay, moving on. The answer is, of course, it produces refraction and reflection. Remember that it has a similar refractive index to the glass. Thus, when the light moves from the glass to the oil, it doesn't get refracted completely away to the side. It passes straight through into the lenses. So, which of the following microscopy techniques is best suited for monitoring 3D cell structures? Is that TEM, SEM, bright field microscopy, phase contrast microscopy, or dark field microscopy? Think carefully here. I'm going to give you guys about 5-6 seconds. Pretty simple. It's in the name, guys. Remember, it's the SEM, the scanning electron microscope. Remember, that's where it uses the microscope. It bounces off the surface. Thus, you can see the entire surface. You can see the 3D structure. You can see the shape. Okay, and our last question. What does gram staining tell us about a bacterium? Does it tell us that it produces endospores? Does it tell us it is pathogenic? Does it tell us it is motile? Does it tell us it has a high lipid content in its cell walls? Or is the answer E? None of them. I'm going to give you guys about 10 seconds here. Okay, and the answer for this one is none of the above. Remember that gram-positive cells have a thick layer of peptidoglycan in the cell wall that retains the primary stain of crystal violet. And the gram-negative cells have a thinner peptidoglycan layer that allows the crystal violet to wash out and thus not retain the stain. So what the stain tells us about the bacterium is what type of cell wall structure it has and what is the chemical makeup of its cell wall to an extent. Okay, guys, thank you so much. I will hope you guys have attended or will be attending lab this week. And then I will see you soon with the next lecture. Okay, bye.